Thanks very much, Tara. Good morning to all of you. By way of introduction, I want to tell you all that my dad is a retired high school biology and chemistry teacher. So I have immense respect for what you do, and also as a mom of a incoming 4th and 8th grader, again, continued respect for everything that what you do. And so I would like to invite you to, in the course of my presentation, interrupt me at will. Stop me if there's anything that's unclear, because I'm coming into this not knowing the background that you all are coming from, and it's probably different for each of you. And my goal, I guess, in terms of the presentation, I didn't change the slides to bring anything to just a very cartoon level. So I'm going to be showing data, and I'll be giving you the sense of the experiments, how we're doing the experiments, what the results mean. But in every case, I may not explain everything on the slide to your satisfaction, or but I don't want to be telling you more than what you really need to know. So I say this as a means of allowing you, if there's something that just you want to know more and I haven't told you enough, please stop me and tell me that. Now, again, by way of introduction, I'm going to be talking about stem cells and epigenetic reprogramming with respect to cancer. And so my introduction to stem cells first is using Calvin and Hobbes. And what Calvin and Hobbes are experiencing here is the morning after a snowstorm. So Calvin says, wow, it really snowed last night. Isn't it wonderful? Everything familiar has disappeared. The world looks brand new. A new year, a fresh, clean start. It's like having a big white sheet of paper to draw on, a day full of possibilities. It's a magical world, Hobbes, old buddy. Let's go exploring. And the connection to stem cell biology is that what we're going to be talking about is stem cells that have the range of development open to them. So it's like Calvin going out and just being able to imagine anything and everything and going out and exploring. And that's really at the start of embryonic development. The cell, the embryo, has not committed any particular cell fates in development. And in the course of receiving instructions, it goes down different paths. And that's what Calvin and Hobbes are doing here. And Calvin says, let's try this path over here. Well, Hobbes says, I don't see a path. And the reason for this cartoon is that this is now really a connection to using stem cells in the laboratory. We're going at this trying to learn about normal development, and we're using our best guesses, our knowledge based upon past observations, but really we don't always know what we're going to be discovering. So we don't always see the path, but we'll make a path. Hobbes is not quite so convinced. And so Calvin has to say, change is invigorating. If you don't accept new challenges, you become complacent and lazy and your life atrophies. So this is really a positive outlook, isn't it? New experiences lead to new questions and new solutions. Change forces us to experiment and adapt. That's how we learn and grow. And so, again, the extension back to stem cells and normal development. So now, in the course of experimenting, normal development, we like it to proceed according to, each person is different, but each solution is the right outcome for each individual. In the laboratory, we don't always know, and we can't always predict the outcome. And sometimes it seems as though we're coming off of a cliff, but it opens up fresh challenges. And so, in the end of the cartoon here, they end up in a snowbank. And Hobbes says, well, the problem with new experiences is that they're so rarely the ones you choose. And Calvin, again, the optimist, he says, I feel smarter already. So in the course of today's presentation, I hope to bring you to a smarter level of realization about developmental biology and why we're studying epigenetics with respect to figuring out when things go wrong, if we're going off of a cliff here, and leading to a pathologic state. So we're studying cancer biology using stem cells. Okay, so stem cell biology. I'm not quite sure what you have talked about in the course of your time here at Harvard, but really a lot of the excitement that stems around stem cell biology is that it's a paradigm for regenerative medicine. We appreciate the fact that there are resonant stem cells in the body, in the nervous system, in pancreatic, perhaps in endodermal tissues, as well as in the blood system. And what we're trying to do in the laboratory is figure out the cues that take an uncommitted stem cell down a path of differentiation and learn about those cues of normal differentiation that allows us to understand the development and homeostatic mechanisms in vivo. So there are two properties of the stem cell that are equally important. So the stem cell has this ability to differentiate into all cell types, but it also importantly has the ability to self-renew. And this is also a really, really important property with respect to cancer biology, is that cancer has cells within a tumor that have this capacity of reseeding new tumor growths, metastasizing. So these tumor-initiating cells are sometimes called cancer stem cells. And so this is going to be some of the focus of my talk and the work in our laboratory. And with respect to the work at Harvard Stem Cell Institute, there has been an effort, so Lee Rubin is a scientist who's a part of Harvard Stem Cell Institute, who's been brought on board to develop a high throughput screening platform that allows us to do gene and small molecule-based discovery assays that allow us to learn about the pathways and manipulate those pathways using chemical inhibitors. So I'm going to now shift into something that you may know a little bit less about than stem cell biology, which is epigenetics. And so I think before we jump into epigenetics, I need to define epigenetics for you. And I think the best way of defining epigenetics, rather than giving you the textbook definition, is by comparing it to genetics. So everybody's, I think, familiar with genetics and Mendelian genetics, where the principles of Mendelian genetics are we have our genome, and we appreciate the fact the genome is comprised of DNA, the A, C, G, and T building blocks. And through the course of development as well as through evolution, mutations can change that primary DNA sequence. And through the process of natural selection, both in terms of populations over time, as well as in our body, there are selective pressures that cause cells and organisms and populations to evolve. And what I'm going to be talking to you about today is changes not in the primary DNA sequence, yet changes that manipulate the expression of genes in the genome. And I'm going to be talking about a chemical modification to DNA called DNA methylation that manipulates a very small, it's simply the addition of a carbon and 3 hydrogens on a cytosine base. Okay, and I'll show you the structure of that in 1 second. So it's a very small manipulation of the cytosine base, but it has very dramatic consequences, the presence or absence of that modification. And what I'll be showing you is the results that we derived from using mouse genetic models, using cancer models in vivo, where we manipulate the DNA methylation state. And what I'll show you is that by changing the methylation state, we both have an impact upon the genetic mechanisms that lead to cancer. So loss of methylation actually can drive mutational mechanisms that promote cancer formation, as well as aberrant changes in methylation can go through this pathway here, an epigenetic pathway. So acquisition of methylation can occur at the regulatory sequence of genes and inappropriately shut genes off. They're required, let's say, as the cell breaks to prevent cancer formation. And so this particular type of epigenetic change, the gene silencing mechanism, can be thought of actually as an adaptive response, like Lamarckian evolution, where it's an acquired change that also is responsive to the environment. And I'm not going to show you actually the whole biochemical pathway that leads to this one carbon, the carbon and 3 hydrogens being placed on the cytosine base. But suffice it to say, there is in our metabolic physiology of our bodies, the food that we eat impinges upon the environment, meaning the pool, the metabolic pool that can drive this methylation reaction. So when you say to your students, or you think to yourself, you are what you eat, this is, in fact, one of the mechanisms for you are what you eat. Yeah.
These are primarily somatic cells, though, not germ cells, so they probably wouldn't be inherited, right?
So there are actually there is some evidence that's emerging within the last couple of years for transgenerational effects of changes in epigenetic information.
So that's very Lamarckian.
It's very Lamarckian.
Yes, yes. So, evolution is driven probably primarily through genetic mechanisms, but that's the dogma. We need to have more data to convince us of the relative balance. What we're learning with respect to cancer models, of course, cancer from the 1970s we learned was a disease of genes and mutations. And so that's really the mentality, the dogma is with cancer genetics. And so what I'll be telling you today is that there's an equally important path to cancer, which is an epigenetic route. And it's not just an emerging field anymore. Within the last, certainly the 15 years since we were developing animal models, there was already data in the literature that, of course, caused us to ask these questions with respect to the animal model. There were changes that were being seen in human tumors, and nobody knew if this was a bystander effect, a hitchhiker effect that was being caused by a tumor having been formed, or were these changes that were occurring and somehow driving the mechanism. And so what I'll show you today is a model that we've developed to preemptively, or several models that we've developed to preemptively change the DNA methylation state on the genome, and then ask, what is the consequent effect on tumor genesis? So, and what I'm going to wrap up with by the end of the talk is a question which we haven't answered yet, which is what we're really very interested in now, which is whether or not stem cells in the body and cancer stem cells within a tumor show a difference in terms of their epigenetic regulation, their dependency on the machinery that catalyzes these events. And the reason being is that some of these pathways, we've shown that they are therapeutically relevant. There's been drugs that have been developed and are actually approved medicines in the clinic. What we don't know now is whether or not there is going to be a long-term adaptive response to those type of drugs that are going to alter, and whether or not there's a stem cell compartment within a tumor that over time might emerge and be a more aggressive disease. And so that's the question that my laboratory is addressing that I'm not going to be able to answer for you today. So the enzyme reaction, DNA methylation, again, occurs at this base, cytosine and DNA. There are enzymes that use a small molecule cofactor called S-adenosylmethionine that transfer the methyl group to the cytosine base. And so the enzyme actually attaches here and transfers the methyl group to that 5-carbon position. Now, in mammals, there are 3 genes that can catalyze this reaction. And so starting off with unmethylated DNA, you can acquire methylation de novo using enzymes that are encoded by 2 different genes, DNMT3A and 3B. So they can put new methylation on the genome. But in the course of cell division, you get the occurrence of a methylated parental strand and an unmethylated daughter strand. And it's this gene here, DNMT1, that is responsible for re-adding the methyl group to the daughter strand. So it's required for maintenance methylation in all cell divisions. So because there's only one gene that's responsible for this, in the course of the studies I'm going to be talking to you about today, most of the work that we've done has been focusing on this enzyme. And I'll show you a little bit of data where we've intervened genetically to manipulate one of the de novo methyltransferases. So the functions of DNA methylation, by mutating these genes in mice, and here I'm not going to be talking about the technology per se for doing this, but if you have any questions, please feel free to ask. We know that mutations in DNMT1, 3A, and 3B are lethal. So it tells us that DNA methylation and these genes are essential for mammalian development. I'll be showing a little bit of data where we've looked at the role of DNMT1 and DNA methylation in chromosome stability, and the results of those experiments tell us that the loss of methylation can promote chromosomal instability, which tells us that methylation normally has a role in maintaining genomic integrity. Okay, I will also be showing you some data that shows that DNA methylation serves as a signal for selective gene silencing. I won't be talking to you about the mechanism of that, but there's a fair amount known in the literature from other people's work that shows that DNA methylation changes can lead to chromatin changes, meaning how the DNA interacts with non-histone. I'm not going to be talking to you about chromatin, meaning the nucleosome structure modifications to nucleosomes, and that is another level of epigenetic regulation. So by speaking mainly about our work, I don't want to leave you with the notion that there isn't much more out there. This is really the tip of the iceberg, and we could spend really, a whole course. In fact, we do spend whole courses talking about this subject. And the last point down here is that DNA methylation changes are an essential epigenetic mark for gene regulation in certain developmental contexts. So I'm not going to be talking about X chromosome inactivation, but I imagine that some of you have heard about this principle. It's a dosage compensation mechanism between female and male mammals. And so similarly to X chromosome inactivation, where female cells have one active and one inactive chromosome, there are about 100 or so genes in our genome that we know about that are expressed from one or the other chromosome. And the selection for that expression pattern determines on the chromosome of origin. So there's some genes that you inherit, that you express only on the chromosomes that you've inherited from your mother, and other genes that you express only on the chromosomes that you've inherited from your father. Okay, and so this process of parent of origin specific gene regulation is called genomic imprinting. And what we have done is created a model for loss of genomic imprinting by mutating this DNMT1 gene very selectively, and I'll be talking to you about the results of that experiment shortly. So before we get into the in vivo phenotypes, I want to tell you a little bit about what's known about the remodeling of methylation. Where is methylation on the genome normally, and how does it change in the course of tumorigenesis? So in the genome, methylation resides at a dinucleotide context, CG dinucleotides, right? And so what this figure is showing is CG dinucleotides in a cartoon form depicted as a lollipop. And if the lollipop is white, it's non-methylated. If the lollipop's red, that's a cartoon showing that it's a methylated cytosine at a CG dinucleotide. So what the cartoon is meant to depict is that there are certain sequences that have a high density of CG dinucleotides, and they're extensively methylated. So these are repetitive sequences that are near structural elements such as centromeres, telomeres, and endogenous parasitic viral-like elements. Okay, so our genome is about 50% of these repetitive elements, viral elements that have come into the genome of different types. And these sequences have a high CG content and are extensively methylated. And it's because the cytosine base, actually the methylcytosine base, I'm not going to show you the chemistry for this, but it can lose an amino group at the 4-carbon position, and when that occurs, it becomes thymidine. Okay, so that chemical reaction turns a methylated cytosine to thymidine, and that's a very common mutation. So there is, you might imagine that if you took the CG dinucleotide and had it randomly stretched across all the DNA in the genome, all these lollipops should be uniformly scattered from one side to the other, right? But because of this mutagenic effect of the methylcytosine, over evolutionary time, it's non-random. Okay, and so where we see residual CG dinucleotides, we can infer a couple of things. One is that it's either selected for or it's non-methylated in the gametes where those sequences, if they're methylated, are going to be prone to deaminating mutations. Okay, and so there are certain promoters of genes, regulatory sequences of genes that have a high density of this motif, the CG motif. And these sequences tend to be unmethylated, and that allows the gene to become expressed. Now, I'll show you in the course of looking at tumor cells, here are the changes. So these repetitive sequences are undermethylated. And because of that loss, because of that undermethylation, because 50% of our genome is this sequence, if you look at total methylation in the genome, comparing a normal cell to a tumor cell, tumors have less methylation overall. Actually, if you look at individuals from young to middle age to old age, there's a progressive loss of methylation over time. So this is a time-dependent effect. And the consequence of this, as I'll show you, is that these sequences are prone to either inappropriate recombination, so this can lead to interchromosomal mitotic recombination events that can homozygous mutations, for example, or it can lead to wholesale losses of chromosomes, losses or duplications. And so because the centromere is required for proper chromosome segregation, changing the structure of the centromere influences the proper segregation of chromosomes. So again, in tumors, we see that there can be certain methylation events that occur within gene bodies, and because of the mutability of the methylcytosine, there are C to T transition mutations. So the p53 tumor suppressor gene, and show of hands, how many people have heard of p53? It's one of the very earliest tumor suppressor genes that was ever described, and it's a really, really important protein with respect to preventing cancer. So about 50% of the mutations that occur in p53 are actually a hotspot mutation that's caused by a deamination mutation. So it's a really important mutagenic mechanism in the course of tumorigenesis. And so the last point here is that there are these regulatory sequences, which are normally unmethylated, that in the course of tumorigenesis, we can see that there are genes that acquire methylation that aren't present in normal cells. And so we had, going into these experiments, a number of questions. One with respect to the loss of methylation, what is the importance of that? The second, what is the importance of acquisition of methylation? When and how, what is the relative importance of each of these changes? And so the results that I'll show you using a few different approaches, I think really show that the loss of methylation and what's actually not shown here is that loss of methylation can even take a gene which is not normally expressed and allow it to become expressed. Okay, and so we have some evidence now where we've caused this inappropriate activation of gene expression. So either inappropriate activation of gene expression and chromosomal mechanisms by undermethylation of the genome can promote early events in cancer. And these hypermethylating events can lead to progressive selective events as well in tumorigenesis. Now, this is looking at one particular model system, which is colon cancer. Now we're looking at brain cancer, and we don't know to what extent the observations we've made in one context are applicable to others. And really, I think every single tumor is going to be something that we have to ask these questions in, because with respect to these different mechanisms, it may be that the order of events of mutation and epigenetic changes is really important with respect to different types of tumors. And so by saying that a certain change is important early and other change is important late, that may have really a defined context for meaning. I think you had a question?
Yeah, I was just curious of if there's any data to support the number of methylated groups across the entire strand versus, because you've got some large groups here and some large groups there. versus, when it just gets redistributed? Is there any data that, correlates the total number in both normal and tumor cells besides just the areas where they're?
So really the correlation is really with respect to total, really reads out only the loss of the repetitive sequences, because the repetitive sequences really dominate the genome. Genes are really a very small fraction of our genome, and our genome is really designed to regulate those genes in a very orchestrated way. And that's really what a lot of the data that's emerging. I don't know if you're reading the science part of the paper, but a couple of weeks ago there was a study that was published, and I saw the press about that said, well, how we define a gene might be changing, because what we see is that there's actually transcription that's all across the genome. And so it's likely true. One idea I think that is emerging from those studies, as well as what we know about cancer, is that these sequences are regulatory sequences, and the loss of this methylation is probably uncovering new binding sites for the transcription machinery to come in and start transcribing. And so the loss of methylation can really relieve so there's probably two mechanisms that the methylation serves on these sequences. One is a structural role to prevent recombination, and the other is a transcriptional silencing role. And that transcriptional silencing role prevents transcription from going out into neighboring sequences. And this is really the emerging idea, which we don't have any data for as yet, but that transcription from these sequences, inappropriate transcription, is causing RNAs to form. And these RNAs, and it's been shown in model systems like single-cell organisms like yeast, that transcription into promoters can lead to gene silencing. Okay, so there is evidence that allows us to build a model. We haven't proven that that's really the model that occurs in human tumors, but I think it's a really intriguing and very interesting idea. So the experimental system in question that I'm going to start off posing is with respect to colon cancer formation, and this is a diagram that was derived in the early 1990s from a group at Johns Hopkins, Bert Vogelstein's lab, where they were studying using a cancer genetics approach, where they were taking normal and various progressive stages of tumorigenesis and asking in a very systematic way about changes in gene mutations and gene copy number. But one observation that they made is that the DNA was undermethylated at a very early stage. And so we were asking in the course of our experiments whether that undermethylation was important to drive tumorigenesis. And so the way that we approached that problem was by taking a mouse that had a mutation which is in the same gene which is mutated in human colon cancer. So this mutation in the gene called APC for adenomatous polyposis coli, when it's mutated in people, gives rise to a familial syndrome where the patients get colon cancer with 100% penetrance. They have to have colonoscopies, and they have polyps that occur in their teens and early 20s. And so a mutation was made in Bill Dove's lab at the University of Wisconsin that mimics the phenotype that's seen in humans. So you get these intestinal polyps. And so what we asked is whether or not if we reduce the methylation status in this mouse model, would we drive tumor formation? That's what we were thinking, in fact. And so the way that we approached this, I told you that mutations in the enzyme that we were studying are embryonic lethal. So we had mice which were wild type, and we had mice which were heterozygous for the mutation, and they're both viable. So we reasoned that the mice that were heterozygous, they have a little bit less enzyme, and if we took a drug which could interfere with the methylation reaction, we asked whether or not, if we started off with 100% versus 50%, if we could titrate the levels down sequentially in both of those groups. And so we then turned to a drug which was developed actually as a cancer therapeutic, which is now, in fact, an approved therapeutic to reverse gene silencing, called 5-azadeoxycytidine. And the way that this drug works is it's a cytosine analog, and it has a nitrogen in the 5 carbon position that blocks the transfer of the methyl group. Okay, so the way it works is it has to incorporate into DNA. When the enzyme goes to catalyze the methylation reaction, it gets trapped in its mechanism, and it's blocked in its ability to transfer. And because it's covalently attached through its mechanism of action, the enzyme has to be degraded for the cell to remain viable. So the protein gets degraded in the course of releasing itself from DNA. Turns out this is a mutagenic event as well. And because the enzyme is degraded, in fact, what we were doing actually worked, is that we were titrating down the amount of enzyme. And so the approach was to take these min mice, which were predisposed to colon tumors, and inject them either with PBS, a non-drug, or with PBS with the drug, and compare mice that were wild type or heterozygous for the mutation for the incidence of these intestinal polyps. So the wild type mice developed a little bit more than 100 tumors, and mice that simply had a mutation in this enzyme, one copy, half the amount of enzyme, had already a 60% reduction in tumor formation. When we took the wild type and the heterozygous mice and injected the drug, we saw a further reduction in tumor formation, where in the case of the heterozygotes with the drug, we saw half of the mice there was actually no tumor formation whatsoever. So on average, there was only two tumors per individual, but half of them had no tumors whatsoever. So we were excited by this, of course, because we had blocked tumor formation. But I just want to remind you of the, sledding down the hill and going off the cliff. We had a certain prediction, and we actually came up with the opposite conclusion. And what that conclusion led us to is the idea that more enzyme and more methylation in the context of colon tumorigenesis is a bad thing. So somehow blocking this reaction is preventing the formation of these large tumors. Okay? And I'm going to be really trying to summarize a number of experiments. So what we wanted to do was figure out how this is working, and we ruled out that it was working through mutational mechanisms using mutational reporters. We ruled out a number of different using candidate gene approaches. Is it working at the level of the tumor suppressor gene that we're studying? And I don't need to get into the details of that, but what we appreciated is that it's likely that there are genes that are being silenced that are dependent on that. And I'll be showing you a little bit more data that is going to lead to that conclusion. So what we then turned to was asking whether or not under methylation, instead of using a drug to do these studies, we wanted to ask, in a drug-free system, can we approach the same question? So Francois Gaudet, who was a graduate student in Rudolf Jenisch's lab, was studying this enzyme, DNMT1, and he happened to make a particular version of the gene that expressed a lower amount of protein than normal. And when he made mice with this small amount of protein, he discovered that these mice came down with T-cell lymphomas. Okay, so the undermethylated mice came down with T-cell lymphomas, and what this is showing is the methylation level on the DNA, taking a look at one of those repetitive viral elements. So if we inactivate the gene and we cut the DNA up with an enzyme which is blocked by the methylation reaction, then the DNA is high molecular weight. If we strip off the methylation, the enzyme can chop it up into small pieces, and that's shown here. So if we take the thymus of this mouse, which has less enzyme, you can see that the DNA is chopped up more in the case of the mutant animal than in the wild type. So it's not chopped up completely, but it is substantially chopped up, okay, by the enzyme. So the DNA isn't chopped up. In the course of the experiment, we can reveal that there's less methylation on the chromosome caused by the mutation. And so by looking at the chromosome constitution of the tumors, what was found is that chromosome 15 was there in an extra copy in many of the tumors, 10 out of 12. And there's a particular oncogene, the c-MYC oncogene, which is resident on chromosome 15, and was then found by looking back at the tumors to be overexpressed. And we know through transgenic studies in mice that overexpression of this gene causes T-cell lymphomas. So a simple idea here then is that undermethylation in the context of T-cell development can lead to chromosomal changes that cause an aberrant gene expression pattern that can drive tumorigenesis. So what we wanted to ask was going back now to the colon cancer model. What happens under methylation here in the context of T-cell development? Less methylation can promote tumorigenesis. And I just told you in the case of the intestine, less methylation blocks tumorigenesis. So we wanted to ask, using this exact same mouse, what happens if we cross this mouse to the mouse that gets intestinal tumor formation? What's the prediction? So I'm going to jump over this and just go to the result. What we found by looking at very, very early tumors, and I can actually show this. So in the course of tumorigenesis, you can actually find really, really early lesions if you do sectioning of the intestine. And so Yasu Yamada, who is a pathologist, sectioned the intestines and showed using an antibody stain, he could identify these lesions, these really early lesions. And the same marker is then present in the tumor. So you can see here's a full-blown tumor and here's a very early lesion. And so he asked, in the context of early lesions versus full-blown tumors in these undermethylated mice, what happens? And it turns out in undermethylated mice, there's more early lesions and less tumors. Okay, so this brings together those two observations. It turns out, and I think I included data, but I'll just jump over it. These early lesions are dependent upon a chromosomal event, just like the T-cell lymphomas. So these chromosomal events are promoted. The loss of change of chromosome number is promoted by undermethylation, and gene silencing is blocked by undermethylation. You require this enzyme to propagate the gene silencing. So in the course of generating a full-blown tumor, the gene silencing mechanism is required. So you can generate the small lesions, but they don't progress. Okay? And so that result is then summarized here, is that undermethylation is promoting this early stage of tumor formation, but the undermethylation blocks later tumor development. Okay, and so we then asked whether or not we could find evidence using a genetic approach by mutating the de novo methyltransferase DNMT3B to ask whether or not DNMT3B, which we were supposing would be driving these gene silencing type of events, asking whether or not it showed any changes in formation of early lesions versus late lesions. And I'm going to just jump over a lot of the data and just get to the punchline here, which is the observation was that there was no effect of mutating the de novo enzyme in the formation of early lesions, but there was selective requirement for the de novo methyltransferase in the formation of late lesions. So the full-blown tumors depended upon having a de novo methyltransferase. So again, consistent with the notion that gene silencing is playing an important role in the formation of these tumors. So we wanted to ask that question more directly, more specifically, by looking at transcriptional changes. And the way that we went about doing that is by using another trick of mouse genetics to manipulate this gene DNMT1 that we've been studying using sequences that are recognizable by a bacterial recombinase called CRE. So these sequences called LOXP sites we embedded in the introns, flanking a couple exons, such that mutation, by expression of CRE recombinase, we would delete these two exons, and that results in no RNA and no protein. Okay? So what we have then is an allele for this gene which is normally expressed, and when we turn on the CRE recombinase, we can selectively delete essential exons that inactivates the activity of the gene. So this allows us to allow normal development to proceed And to take away the enzyme, we know that the gene is essential for development, so we take away the enzyme, and then we ask what happens if we take away the enzyme after development has proceeded. And we then reasoned that we could generate enough cells of a defined cell type that would allow us to ask this question about the role of DNA methylation in regulating gene expression. Okay, so I'm going to jump into now a little bit more background to give you the notion for the selective model that we were trying to occur. So we created this mutation in DNMT1, and it turns out, and I'll show you a little bit of this data, that when we mutate this gene, it's required in all cells. So cells die, and we're able to, I'll show you a little bit of data that we were able to prove that the gene is required for gene silencing. And we wanted to then direct our attention to a defined set of changes with respect to cancer. And those are these genes that I talked about earlier, the ones that are inherited differentially between your mom and your dad, okay, these imprinted genes. So before I get into the details of that model, I want to give you a little bit more background for the rationale and the importance of genomic imprinting. Okay, so we can take a little bit of a breath on data at the moment. So genomic imprinting is observed in a number of pediatric disorders. These are rare disorders that occur in about 1 in 15,000 to 1 in 30,000, depending upon the different syndromes. These are kind of more frequent or less frequent. So these pediatric syndromes, you can see, have phenotypes that are cancer predisposition, fetal overgrowth, metabolic defects, as well as mental retardation, more metabolic defects. So there's metabolism, mental function, and overgrowth and cancer. Now, there's loss of imprinting, meaning these genes that we know have this monoallelic parent of origin specific regulation have been observed to be deregulated in many forms of human cancer. And so we wanted to specifically address this question: what is the role of loss of imprinting in tumor formation? Is it, again, one of these bystander effects, or is it causally important? And so in order to do that, we wanted to be able to selectively regulate the set of genes that are regulated by imprinting. And to do that, because they're regulated through DNA methylation, we had to do some tricks, and I'll show you about those tricks in just a moment. So another area of importance is that assisted reproduction is actually leading to an increased prevalence of these types of disorders. And so with respect to stem cell biology and the use of embryo culture, it's something that we have to be aware of, is that there's data, epidemiologic data in the literature, that says that the way the technique that's being used, embryo culture, is leading to an increase in these type of syndromes. And so it's something that we need to be aware of, that the manipulation of embryos in vitro may not be optimal with respect to retaining normal epigenetic status. Okay, so the establishment and maintenance of imprinting is described in this slide, where we have an egg and a sperm shown here, and there is, during the course of gametogenesis, a mark that's differentially placed on the paternal versus the maternal chromosome. For simplicity, I'm only showing one pair of imprinted genes here. And in this example, there's methylation placed here, that in the course of looking at somatic cells, when this imprinting is normally maintained, you see that there's a paternally encoded growth factor which is expressed, and it's because of this modification, there's a partner gene which is expressed at the expense of the IGF-2 gene on the maternal chromosome. Okay, so because of this epigenetic mechanism laid down during gametogenesis, there's a parent of origin specific regulation of a gene that's a known growth factor.
Does that mean the female growth factor is shut off in in the egg?
So
I don't see little stars next to it.
there's,
Is that-
right, so there's no expression of this gene.
But is that because it's been imprinted up there?
Right, so the imprinting actually occurs, I didn't put in a slide that shows exactly how this works. This is a chromosome pair that resides in about 100 kb on chromosome 7 in mouse. It's syntenic in human. And what happens is there's methylation that resides differentially on the paternal versus maternal chromosomes. That's at the 5' end of this gene called H19. The H19 simply encodes an RNA. There's an enhancer that's distal to the H19 gene that can regulate H19 and IGF-2. If you shut down H19, this enhancer can see IGF-2 and drive it. Okay, so it's not a direct regulation of methylation of the IGF-2 promoter, but it is regulation in cis, where the methylation here is actually required for expression of IGF-2. Okay? And so the other point to this slide that I want to make is that because in the germline development, in order to have proper reestablishment according to the gender of the individual, you have to erase the imprinting on the chromosomes in early germ cells and reestablish them during gametogenesis. Okay, so in somatic cells, imprints need to be maintained. If they're not maintained, there's no mechanism to reestablish that information. They need to be erased during germline development and then reestablished during normal gametogenesis. And so, because of this developmental regulation of this epigenetic mechanism, what we did was we reasoned that if we could, in a post-fertilization state, and what we used was an embryonic stem cell, manipulate the epigenetic information to selectively eliminate imprinted sequences, we could then look in somatic cells derived from those ES cells. So what we do is we study this in the context of ES cells, or mice derived from the ES cells. They're chimeras that are partially ES cell derived and partially host derived. If we take those mice and we breed them, those manipulations that we made get fixed. But it serves as a useful control, because anything that we observe in terms of a phenotype in these animals, if we breed them, we can show that it's reversible on germline passaging. Okay, so there's one more bit of information, background information that I want to tell you about, is that the history of genomic imprinting, I'm going to summarize it and give credit really to Davor Salter and Azim Sarani, who, using nuclear transplantation, established that the maternal and paternal pronuclei were non-equivalent. So I would think that in the course of your time here at HSCI, you're learning that nuclear transfer allows us to take a somatic cell nucleus and reprogram it and generate a fully undifferentiated embryo that is fully capable of going through the normal stages of development. And what I'm telling you now is that you can reconstruct an embryo selectively using two paternal pronuclei, two haploid genomes that are both derived from dad, and two haploid genomes both derived from mom, and they don't develop normally, okay? So if you take a normal somatic nucleus where all the information is intact and that imprinting was intact, you can direct the formation of normal stem cells. You can direct the formation of stem cells using this technique, yet the development out of those stem cells is not normal, okay? So two male nuclei, two paternal nuclei rather, when you study the development, you can see good development of extraembryonic tissue and poor embryonic growth. And two maternal nuclei generate poor extraembryonic growth. And the embryo can form, but once it depends upon the placental function, then it stalls and stops developing. You can take these same embryos and derive ES cells and use the same approach that we're employing in our study, which is to look at them in the context of chimera formation and say, if you mark these cells, where can they contribute? That tells you something about the developmental potency of the cells. What types of tissues can they direct? And you can see that the paternal genome can direct the formation of muscle, mesodermal tissue, but there's very low contribution to brain. Whereas the maternal genome, there's actually very good contribution to neuroectoderm and low contribution to mesodermal tissue. So in fact, there's really a balance that occurs. Yeah, you're snickering. This is the brain economy. Yeah, you say this to your students, they'll get it too. So we know that this is due to the fact that there are, again, these 100 or so genes that have a parent of origin bias. And there are certain genes that are maternally expressed that are really required to direct the formation of the brain. And that there are a number of paternally encoded growth factors, in fact, that are required and essential for regulating fetal size. And so there's a hypothesis actually developed by David Haig here at Harvard that there's a conflict between paternal and maternal interests, is that from the mom's point of view, she wants to be able to nurture her individuals and survive childbirth, whereas the dad, wants to be able to see his progeny, be best fit. In the case of, one female with multiple males, let's say, if his offspring can survive, his genome has a better chance of surviving and being propagated. So the paternal genome wants to have more, robust, larger, fit offspring, and so this is perhaps at the expense of the mom, who, if this goes unchecked, the babies get larger and larger and larger, and so there has to be some balance. And so there is, in fact, some of the data that I'll show you is consistent with this notion, is that the maternal and paternal genomes are regulating fetal size. Because these are growth-promoting mechanisms, they're likely also important and operative mechanisms in terms of promoting cell growth in cancer. Okay, so I'm going to summarize a number of studies here and get back to this point that I was mentioning earlier, is that by mutating this gene DNMT1, what we observed are two things. One is that the loss of DNMT1 in embryonic stem cells had no effect on the self-renewal capacity of these stem cells. It selectively blocks differentiation, but it doesn't block the self-renewal capacity of ES cells. Okay? So somatic cells die. If you take away this enzyme from a differentiated cell, they require it. If you take it away from a stem cell, it's not required. So here's a really interesting difference. And again, something that we're studying with respect to the importance of this gene in cancer stem cells. Because the gene is required, we've turned to another clever approach to generating the loss of imprinting model. And I'm just going to jump over this. This is really a transcriptional experiment. This was the data. The slides are a bit out of order. The data that we generated to ask the question whether methylation had a role in gene silencing. And the way that we approached this question was to look at the expression of genes in the absence of this enzyme, DNMT1, using microarrays that can look at the expression of thousands of genes in parallel. And what we observed is that loss of methylation caused the overexpression of about 10% of the genes in the genome. So, a really dramatic and profound effect. Now, because there's such a profound effect, we needed to have a more clever approach to selectively manipulate just the imprinted gene. So in the course of looking just at loss of function DNMT1, we saw imprinted genes being deregulated, but we saw a whole host of other genes, and this is an incomplete list. So the way that we developed the loss of imprinting model was not just inactivating DNMT1, but turning it back on again. Because we knew that passage through gametogenesis was required for establishing normal imprints, we reasoned that if we took away DNMT1, we would eliminate the imprinting methylation, but if we turned it back on, we would create a cell, because the de novo methyltransferases are still intact. If we provide the maintenance methylation activity again, those cells have the ability to proceed through normal development, and what we can ask is, what is the consequence simply of eliminating the methylation of imprinted loci? And so we did this by creating a separate mutation in DNMT1 which is a mutation that we can remove. Okay, so this gene, this allele I showed you the cartoon of, where we can take away the essential exons. Here we put a stop sequence within the gene that causes the gene not to be expressed, and we can kick this out with a different recombinase to turn the gene back on again. Okay, and because we have these two different recombinases, we can reverse the order of them, and in the course of doing this, we can generate a control cell which has the exact same genotype, but these intermediates are what are really important. So the intermediate here is demethylated, and the intermediate here is wild type. So what we have is this transient demethylation in these cells that it doesn't occur here. We result with cells that are genetically identical, but have only a very small subset of differences in the epigenetic information, the DNA methylation. And so I'm not going to show you all the data, but the repetitive sequences, the half of the genome that we were talking about, those become remethylated when we do this, but the imprinted sequences remain unmethylated. And so I have simply an example here where, again, these filled in circles show methylation, and imprinted genes, in this case, here's an example called IGF2R. There's methylation that occurs here on the maternal chromosome that doesn't occur on the paternal chromosome. And so you can see methylation here, not here. When we carry these cells through the transient demethylation, we lose all of that methylation, okay? And so the approach is to take these altered cells and to create a chimeric embryo and study the formation either in fetus or in adult animals. And to cut to the chase, what we see in adult animals is that these chimeras develop tumors in the liver, in the intestine, and in the white blood cells, leukemias and lymphomas. The latency of tumor formation is about 9 to 12 months, and we've shown that the tumors are derived from the injected ES cells. Okay, and more also importantly is we've shown that tumor predisposition is reversed on germline passage. Okay, so I'm not going to show you the data. This is just a marker, the imprinted methylation status, to show that in the tumors they have the loss of imprinting status. Okay, so we've gone on to mechanistically ask, how is the loss of imprinting leading to tumor formation? And we've made a couple of observations. One is that these cells early in development appear to be immortalized spontaneously. They don't require oncogenes, they're already immortalized. And if you add simply one oncogene, they form a tumor. They can form a tumor in the context of a cell culture assay called focus formation, or if you take those cells and you inject them into a mouse, they form a tumor. And typically, with a mouse cell, you require 2 oncogenes to do that. So this loss of imprinted methylation is taking the place of one oncogenic hit. And so we've shown by looking at some candidate imprinted genes, an interesting trend is that the paternal genes appear to be overexpressed, and the maternally imprinted genes appear to be underexpressed. Okay, and we don't fully appreciate the complete basis for this, but it appears to be a trend, and this may also be consistent, again, with this notion of genomic conflict between the two parents, is that the paternally expressed genes being overexpressed may be those which are promoting tumor formation. In addition, it may be the loss of maternally encoded genes which are serving as the brakes. And so Guanwen Wang, who's a postdoc in my lab, wanted to ask this question, and asking it, instead of forming tumors in vivo, asking if we could use fetal development, fetal growth, as an earlier endpoint in chimeras. And so he, along with Juliana Brown, looked at the formation of chimeras at 13.5 days of development. We can see that the loss of imprinting chimeras are larger, statistically significantly larger. And because of this overexpression of this paternal growth factor and silencing of this gene called IGF2R, and what I didn't tell you is that IGF2R, the purpose of it is to downregulate the function of IGF2. So it's a maternally encoded receptor that's a sink and takes IGF2 out of the circulation and doesn't allow IGF2 to have a strong growth promoting effect. So not only do we have overexpression of IGF-2, but we have loss of a negative regulator of IGF-2. And so what Guanwen asked is whether or not if he could turn IGF-2R back on, if he could reverse this growth phenotype and reverse the cancer predisposition. And so the way that he went about doing that, and I'm not going to show you any more details than just this cartoon, was he took out this regulatory sequence where the methylation is targeted to on the chromosome. And by deleting this sequence, it actually causes this allele to become re-expressed. And I'm not going to show you that data, but I'm just going to show you the results of the experiment that, in fact, show that here is the size of control and the loss of imprinting embryos. You can see again the shift up. And now if we look at the size of chimeric embryos when we reactivate this receptor, which downregulates the paternal growth factor, we re-establish reasonably normal, statistically significant normal embryonic fetal growth. And so what Guanwen is going on to do now is ask whether or not cells from these embryos also have whether or not they reverted the immortalization phenotype and reverted the ability to interact with oncogenes to cause transformation. Okay, so I've gone on quite long, and so I guess I don't want to take up too more of your time, but just to mention really kind of the last project in my lab without going into too much detail is to get at this question of a difference between the normal cells in a tumor with tumor stem cells with respect to their dependency on this enzyme DNMT1. So we're asking, what is the role of DNMT1 in brain tumor stem cell self-renewal? And the way that we're approaching this question is by taking a mouse model which is predisposed to brain tumor formation. It's a pediatric brain tumor called medulloblastoma. Human medulloblastomas have been shown by a group in Canada to be enriched for a cancer, a tumor-initiating cell that's marked by a protein called CD133. It's a cancer stem cell marker. And so what we've done is to take a mouse which is predisposed to medulloblastoma formation that's caused by mutations in two tumor suppressor genes. One is called the patch gene. It's a negative regulator of an important embryonic regulatory pathway called sonic hedgehog. So patch mutations on their own cause medulloblastoma formation at low penetrance. And by incorporating mutations in the p53 tumor suppressor gene, the latency of tumor formation and the penetrance, so they get tumors faster and with high penetrance. And so the complication is that it's a difficult mouse strain to work with, and what we want to do is manipulate it genetically. And so what Juliana Brown did, Juliana was actually a grad student at Doug Melton's lab here at Harvard. She worked in this building. Juliana isolated embryonic stem cells from this tumor-prone animal, and then asked whether or not we could make chimeric animals using the disease-prone ES cells, and asked whether or not that would have the same phenotype as mice derived by breeding. And I'm not going to go into all the gory details that prove that it's true and it's a good model system, but I think the picture tells everything here. So the red is the chimeras and the black is the mice derived by breeding. And I think the other telling point here is that look at the number. It took a long time to generate 12 animals here. It took a few days of injections, well, more than a few, but a couple handfuls of days to generate these data. And so it's a really nice model system where we have disease-prone ES cells, where we can generate experimental animals at will. And because we can genetically manipulate ES cells, we can change the epigenome. We can directly ask the question of the importance of all these genes in the formation of tumors. And the last point that I want to show here is that because these ES cells, this gene is marked with a LAC-C marker gene, we can see the contribution of ES-derived cells in vivo using an XCal stain. And because they have a mutation in p53, we can use that genetic difference to show that in the cerebellum, at the point where there's tumor formation, these tumors are completely derived from ES cells. Okay, so this is a marker of the host DNA, and in the tumor, we can see completely ES cell-derived DNA. And so this now allows us, what Juliana is doing is to get at the notion of cancer stem cells. She's trying to ask whether or not these tumors form from one or many cells by marking them with differentially colored markers, and ask whether or not do the tumors form with multiple different colors or from a single color, as well as introducing the mutations that I described for you today to ask if we manipulate this pathway, how does it influence the formation of tumors? Is there a difference between undermethylation at different stages of tumor formation? And so that is the experimental game plan. And the last point that I want to mention is that a couple of medical students in the lab, Anup Patel and Shui Lin, are asking a very similar question in the context of the formation of human brain tumors. So we have a collaboration with Scott Pomeroy's lab at Children's. Scott is the chief of neurology, and he's been studying human medulloblastoma for many, many years. It's a really, really important resource. He's been banking tumors and deriving cell lines from these tumors. And so what Anup and Shui did was they took this marker for cancer stem cells, CD133, and looked at the panel of tumor cell lines and found one which is a very, very high prevalence of this cancer stem cell. And so what they've done is taken this cell line and shown that they can transplant that into the brain of a mouse. By marking these cells with a GFP marker, you can see GFP fluorescent tumors forming. And so now what we're doing is we're trying to take away the DNMT1 gene and ask, what is the role of DNMT1 in the context of human cancer stem cells in the brain for formation of tumors? So of course the way scientists approach their problems is we don't know the result of that question. And so the way that we're starting is by taking a protein where we have a very strong prediction. And so the gene BMI1 is known to be important for normal neural stem cell self-renewal. And it's also known to be overexpressed in medulloblastomas. So the first experiment they've done is to generate genetic vectors that allow us to eliminate the activity of BMI1, knock down this gene in these cells using, again, GFP-tagged vectors, and taken these cells and put them in the brains of mice, and found that that blocks the formation of tumors. Now this is a really preliminary experiment. We only have one mouse of each case, so I'm showing you really early data, but is supporting the notion that this protein, which is important for normal stem cell self-renewal, is likely also important for cancer stem cell self-renewal. We have an assay system where we can ask the same question for all these genes in the pathway. So I'm sure that I've given you tons of information, but I want to kind of sum up with a couple smaller conclusions, and that is that DNA hypomethylation, undermethylation, can promote or suppress tumorigenesis, and this depends on the oncogenic mechanism. Okay? And then I showed you a second model that we developed where we selectively altered the imprinting status of cells, and loss of imprinting appears to predispose to cancer and provides really, I think, the first example of a purely epigenetic mechanism for promoting cancer. So there's no mutations that can be invoked in that model. Now, I want to leave you not with just not just the simple conclusions, but to leave you also with the broader questions that we're faced with. And that is really, I think, really with respect to everything that we do in the laboratory, we're constantly asking ourselves, well, to what extent do rodent models reflect human cancer? So I told you that it took 2 oncogenic hits to transform rodent cells. It turns out that more than 5 hits are required for human cancer, and we don't know how many epigenetic hits are involved. So this is part of the reason why everything that we do, we want to recheck what we do in an experimental platform that allows us to extend our observations back to human, because the notion of our experiments is to develop therapeutic proof of concept experiments. And many of these, if you look at drug discovery, not every single good therapeutic target ends up leading to a drug. Actually, very, very few do. And the reason for that is that the preclinical models aren't always predictive of the response in humans. So I think it's really, the burden is on the experimenter to really prove that what you observe translates, okay? And then I left you, I think, also with this broader question of, well, studying epigenetic regulators And what I didn't really drive home probably is the notion that epigenetics is important for stem cell self-renewal. But this is really something that will be a prevailing theme. I hope that you're hearing from other people that are speaking to this in the course of learning about nuclear reprogramming. Are you getting talks from anybody else?
You're the first.
Okay.
On the genetic reprogramming. I think this
Okay.
is the first epigenetic
Okay.
talk around that.
Yeah, so, if you have any questions in the course of your time here, Tara also has my email, and so I'm really open and welcome to taking any questions that emerge both from my talk today, but also, if you find that through the course of being here that you have questions that haven't been resolved, that's something that I'm very open to, having a continuing dialogue with the group of you. And so I want to close by thanking a number of people, the group in my lab, of course, that works really hard to carry out the experiments. My postdoc was in Rudolf Jansch's lab at the Whitehead Institute, and the loss of imprinting model and all of the early studies I talked to you about were carried out in his lab while I was a postdoc. I've been very fortunate to receive funding from a number of places and have a collaborator in Austria who was a former postdoc in the Janisch lab who generously provided a vector for some of our studies. And I also want to thank a number of my mentors at Children's Hospital. Actually, I should mention, so Tucker Collins was my department chairperson, and in January he was diagnosed with a malignant brain tumor, and he died exactly a month ago. So what we've been doing since the time I set up in my lab has become personally relevant. Not that it wasn't before, but it's become increasingly personally relevant. So with that, I thank you for your attention, and again, I will take any questions that you have.
IVF might be associated with cancer and some of these things that you've shown in your research.
So I should tell you, the connection is to the neurologic syndromes, Angelman's. There is actually, I think, also a paper on back with Wiedemann and Wilms tumor. But the first observations, I think, that were published were with Angelman syndrome, which has a mental retardation phenotype.
Okay. Autism is increasing in frequency. Is there any connection to IVF, perhaps, or no?
I don't know if autism and IVF have been looked at directly. Autism, what I didn't include are there's a whole epigenetic component to neuronal development. And autism, clearly, it's a spectrum disorder as well. There is a disease called Rett syndrome, which is caused by mutation in a methyl DNA binding protein called MECP2. There are other epigenetic regulatory proteins. There's a protein that regulates histone methylation, so another epigenetic pathway that is an X-linked mental retardation disorder. And so there's increasing and mounting evidence that epigenetic mechanisms are important in neuronal development that probably play out in a whole host of cognitive function disorders. So there's overexpression of DNMT1, the protein I was talking about today, in schizophrenia, and the exact mechanisms and modes of action completely remain to be discovered. But I would say, with respect to mental retardation and cognitive function, the data now look very similar in the literature to the type of data that we were looking at in cancer biology 15 years ago when we set out to address the questions of the importance of cancer, methylation, epigenetics, and cancer. So, if we use that as a guiding principle, I think there's a lot of opportunity of studying these diseases as well. And I don't know that embryo culture, I think the increasing rates, if you will, of autism may be kind of like crime statistics. When you're looking for it, you can find it. And so crime statistics can go up. As we get better at refining what we call autism, when you have better diagnostic criteria, you can say it more definitively, you get a better diagnosis.
Are you developing any models to look at hypermethylation?
There's a postdoc who's in Rudolf Janish's lab now who's addressing that specific question. And so what he has done is developed an animal model where he's ectopically driving too much of the de novo, one of the de novo enzymes in the intestine. And so he asked that question that you're begging, right? Is what happens if you drive too much of that enzyme? If you take it away, you know it's required. If you drive too much of it, do you get more tumors? And the answer is yes.
Recently there was something about using fibroblast mouse, fibroblast cells to take on an embryonic stem cell nature.
Yeah.
And I mean, I see the germline as already being in a sort of a low methylation state, you know, and so there's some reprogramming that goes on there. But when you talk about the development of an entire organism, I imagine there's a tremendous amount of methylation that's occurred. What proteins or enzymes or what really is resetting that clock or resetting that genome?
The first observation was reported last summer, and it was a group in Japan that carefully looked at overexpression of transcription factors that were known to be expressed in pluripotent early embryos. And they overexpressed them in combination and selected out a combination of 4 factors that, when overexpressed together, could take a somatic cell into a more embryonic-like state. Okay, and in the course of doing those experiments, their first paper, they could show that they could get contribution to all the lineages in the embryo, but they didn't establish germline potency. In the most recent papers, again, this group from Japan, Rudolf Janish's lab at the Whitehead Institute, and Konrad Hochelinger, who was a postdoc in the Janisch lab, who is now part of HSCI, all 3 groups reported the same observation, is that you can take these 4 transcription factors and drive them, and by carefully working out the selection methods, improving the selection methods, you can generate ES-like cells, where they call them IPS cells, for induced pluripotent state. And so these transcription factors, it turns out, 2 of them are known transcription factors that are pretty famous for being genes that we knew were important for pluripotency already. So OCT4 and NANOG. So these are transcription factors that are required for early embryonic development, are known to be essential for maintaining the undifferentiated embryonic stem cell state. And then there are 2 more proteins that came out of this experiment. One is c-MYC. I told you about c-MYC today. c-MYC is an oncogene. And the other protein was called KLF4. And how all these proteins in concert act on a differentiated genome and serve to reprogram it to an embryonic-like state is the $64 million question. That's what everybody wants to know, because nobody wants to put, first of all, transcription factors into a cell. You don't want to put oncogenes into a cell, but you want to understand how they're acting so that you can generate another type of molecule that you can transiently put into the system and generate that same defined result. You want to be able to take somebody's skin cell, throw some factors on that aren't going to cause cancer later on, and generate that induced pluripotent state. So I think that's, it's a beautiful set of experiments. Konrad, who is at MGH now, showed that the genome in these induced pluripotent state cells is epigenetically reprogrammed, looking at X chromosome inactivation in a very normal way. So they basically can reset the clock and go through development in a normal way. It's really beautiful papers, but it's very early days with respect to how it all works and how that observation is going to influence regenerative medicine. But it's a very exciting time.
Sure.
Is there data that if the food we eat has methyl groups in it, some of it does,
so
or more or less?
folates, folic acid is one methyl source, and
That's
it turns
the
out
one for
so
you need your-
neural
make sure that pregnant women,
Correct.
is that?
So it turns out in 1998, the FDA mandated that there's actually folic acid supplementation in wheat flour. The flours that we use that are enriched actually now have folic acid. So in the last 10 years, you've been getting more folic acid than you realize. And this was mandated. And there's, the March of Dimes has done a study. There's epidemiologic data that it may actually be beneficial to neural tube defects. We've got no idea if there are other consequent effects to that. But really, again, I think once we appreciated the fact that folic acid was a key dietary component, all women, who are planning pregnancies, certainly their obstetrician puts them on prenatal vitamins even when they're trying to get pregnant. Because it turns out that very early window, before you know, is a critical window for folic acid. So folic acid and vitamin B12 are two of these cofactors that are required in the 1-carbon biosynthetic pathway that leads to the formation of this cofactor S-adenosylmethionine, which is required for all transmethylation reactions. Okay, so this was a project that I did with our public relations department, and we had some computer graphics and animation people that we worked with as well, who took our ideas and turned it into the product. And let's see where. So if you go to this, the idea is that you're entering into a virtual laboratory and you're given a petri dish and you're given the ability to manipulate stem cell differentiation. So you start off with uncommitted cells, and then you're given factors that allow you to direct development down the different embryonic lineages. And so on the right-hand side, we have a descriptor of the feature and also a descriptor about the cells that happen to be growing. So here we have embryonic stem cells that are growing, and this tells you something about the embryonic stem cells. If you click on Enlarge Tree, you can see here are all the different types of cells that you can generate in the course of this virtual experiment. So you start with embryonic stem cells, and you can proceed down the 3 germ layers, ectoderm, endoderm, and mesoderm. And there are a number of endpoints in the differentiation experiments that are different types of cells that are potentially therapeutically useful, and we say something about these cells at each intermediate point. And so if you look at the color of the different arrows, that's actually the code. This is a cheating way. So this is the grown-up's way of going, oh, if I do purple, then orange, I can get to pancreas. Okay, so we can try that. Actually, neurons are the coolest. They're my favorite. Sorry, so let's do blue, then yellow, then green or purple. So if we put blue on here, you can see that they start to differentiate. And cells, when you grow them in a petri dish, if they grow too dense, we have to subculture them into new dishes, and so the computer does that for you. You don't have to make media or anything. And, okay, I've already forgotten. So if you select a factor that isn't the right thing, it says, right, I remembered actually, but I just wanted to show you. If you pick the wrong thing, you can see that the dish becomes the color of the factor that's on them, and it tells you, it's not doing anything because it has no effect. So here, once you change it to yellow, now we get neural stem cells forming. And so if you differentiate so neural stem cells can still divide because they still have the property of self-renewal, but they will also spin off neurons. Neurons stop dividing, so they just sit there. So this is something that, if you're trying to teach stem cell biology, you can probably use this as a tool to captivate somebody's attention for at least a little bit of time. And my son actually made some decent suggestions that got incorporated. So yeah, I thought I'll tell you Virtual Stem Cell Lab, and I can also send a link to Tara if that's something that, but it's easy to find on the web.
Question about cell differentiation. When it finally gets to the point where it's ready to differentiate, where's the point of no return?
The question that you're asking is basically cutting to the core of regenerative medicine and stem cell biology, right? And so in the course of normal development, you start off with a genome which has the full range of possibilities. It can form, you know, if we take this tree as an example, an embryonic cell can go down all 3 germ lineages, and it can contribute to all different types through progressive differentiation into all different tissues. As a cell proceeds down one of these lineages, its possibilities can still remain open, but it's restricted. It can't form these lineages, but it is open to these, okay? In what the reprogramming experiments tell us is that we can drive the clock backwards. We can direct this clock backwards. In the course of normal development, what you're asking is an answer to a question that we don't really know, is whether or not cells at any point in time make a decision to go backwards on their own. Are there cues in vivo because stem cells are being depleted? Are there normal cues where to restore homeostasis, the body says, we don't have enough of these type of cells, and what we need to do is de-differentiate and make some, otherwise we're going to just run out. There's no evidence that suggests that occurs. In the course of tumor formation, these cancer stem cells give us a snapshot of a stem cell population, but we don't know what's the chicken and the egg. We don't know, is the cancer forming from a cell which was a stem cell, and because of that, it retains this ability to self-renew as well as differentiate and grow a tumor that can metastasize? Or in the course of tumor formation, do these oncogenes reprogram the cells and de-differentiate them into these more malignant cell types? So this is really the crux of the question that we're interested in with respect to cancer stem cells. I think it's likely that it's a pathologic state. Whether or not it's a normal state, I think it's a really open question. There's no evidence to suggest that in the course of normal development and aging, the body resurrects homeostasis by building new stem cells. But that's one of the goals of regenerative medicine, is to figure out if we can reverse that clock in some way. So we think there's proof of concept experiments using these gene-based approaches that you can do it using genes. Okay, and the question is whether or not we can do it with small molecules. And if we can do that with small molecules, then the next question is, can we take those small molecules and will they have any therapeutic value in the organism? Do you have to take somebody's cell and put it in a petri dish and de-differentiate it to remake cells to transplant them back into the person? Or can you generate a molecule that can be used to go back into the person that's therapeutically beneficial? There's reason for thinking that those therapeutics might work in vivo because there are growth factors that regulate the pool of hematopoietic stem cells, and those and by regulating blood cell development, those types of factors are being used as supplemental therapies to help patients respond from chemotherapy, for example. Okay, so chemotherapy, one of the side effects is depletion of stem cells. That's why you get hair loss, why you have GI effects, because these are tissues that have a lot of stem cell and the property of cell turnover, and so they're the hardest hit by these drugs that hit dividing cells. Okay, and so the question is whether or not you can find ways of protecting those cells that are therapeutically relevant and ways of, in a beneficial way, reversing the clock, so to speak, and regenerating a stem cell pool. So great question. Again, thanks for your attention. Any further questions